Respected director, sir. So now I would like to introduce our today's expert speaker, that is Dr. Sunaina. So Dr. Sunaina has completed her PhD in neuroscience from University of Turin, Italy, and currently she is a postdoctoral fellow in the same university, and she is working on the cerebral cortex, and her areas of interest are intellectual disabilities. She has having a lot of articles and review and research articles in reputed journals, and she is having 6 years of 6 years of experience in research. And today she will talk about investigating the impact of mGluR5 PAMs in mouse models of CKL5 deficiency disorders. So I welcome you again, Dr. Sunaina, and now over to you for giving your talk.
Thank you so much for kind words, also to the director, sir, and to Rohit, sir. Thanks a lot. It really means a lot. Thank you so much. So now I will share my screen. Okay, I hope you can see my screen.
No, it's not visible still.
No, okay.
Yes, now it's visible.
Okay. Okay, so you can see my slides.
Yeah, we can see. You can proceed.
Okay, can you see the change?
No, not. Slides are not being changed.
Okay, let me see. Is it okay now?
Just click on the second slide in the left bar.
Oh, sorry.
You can click on your second slide, Sunaina.
Okay, but actually from here I cannot change.
Yeah.
You can click.
Click on the second slide.
Double click.
No, probably I can't. No, I can change from here. I can go to my slides where I can change, but I don't know if it's visible.
You can use the cursor. So through the mouse, if you can using the laptop, so you can use the mouse and click for the next slide.
Yeah, I'm trying, but
Sunaina, if you are able to see the thumbnail in the left bar,
yeah,
then click on the next slide.
Click on second one.
Click on the second one.
let me try to share again. Now, can you still see my slides presenting?
No, it has gone. No, it is not being.
Okay, let me try. Let me try again. I try to reconnect in another way. Maybe now it will work.
Okay.
No. Can you see, or you can see the other screen? No, I think not this one.
Just open your presentation in a tab and then connect.
Yes.
Yes, it's visible now. Try
You
to
can
change
see
now. Try to change. Yes, now it's changing. Now
it's fine.
Okay. Yeah, it's fine. You can proceed.
Okay, so I can start. You can hear me well?
Yeah.
Okay, so as Dr. Rohit sir told that my work is entitled as investigating the impact of mGluR5 PAMs in a mouse model of CKL5 deficiency disorder. As he told you that I am working in the lab of in a lab in the Department of Neuroscience. So I'm working with Professor Maurizio Giustetto, who is working from last 20 or more years in the field of cerebral cortex. And in our lab, we work on genetic disorders, a few genetic disorders. To start today's talk, I want to introduce a little bit about autism spectrum disorder, which is a highly prevalent class of mental disorders that affect the development of nervous system, which leads to abnormal brain function. And this disease is typically diagnosed within or even before 3 years of life. The effect of this disease tends to last for a person's lifetime, and it has a high degree of symptom severity, as the child with autism do not behave properly. They have abnormalities in the speech, they have deficits in social interaction, they don't make eye contacts, and they have intellectual disability, they have cognitive impairments, and there are changes in the sensory and motor functioning. In the worldwide, it affects around 2.6% of world's population, out of which almost 4%, almost 4 times males are highly recognized as compared to the females. This disease is broadly classified into two forms, that is idiopathic or non-syndromic form. On the other hand is syndromic form. The idiopathic or non-syndromic form is caused by combination of various different genetic mutations, and it also involves various biological, psychosocial or environmental factors. So basically the etiology of this form is not well known. On the other hand, the syndromic form is caused by highly penetrable single gene mutations. So mutations in a single gene can cause this type, and which is actually a good thing, because identification of etiology is really crucial to develop the treatment, to look for the medicines and other things. And which is really very challenging in case of the idiopathic form, because the origin is really complex and we don't know what to look for, what are the mechanisms that are altered in this disease. Whereas studying this syndromic form really makes it possible, because by looking for the molecular mechanisms or biological pathways that are interrupted or that are disrupted in the syndromic form, can provide insight also for the idiopathic form, so in the ASD as a whole. So I want to here highlight few genetic mutations that leads to syndromic forms, such as FMR1, for example, in Fragile X syndrome, UBE3A, which causes Angelman syndrome, TSC1 or 2, which causes tuberous sclerosis complex disease, and SHANK3, which causes Phelan-McDermid syndrome, and MACP2 and CKL5, which causes Rett syndrome and CKL5 deficiency disorder. Today, I want to talk about the CKL5 deficiency disorder, which was previously known as a variant of Rett syndrome. The classical Rett syndrome is caused by mutations in MACP2 gene, which is located on the X chromosome, and the previously known as Hanifel variant, which was caused by CDKL5 mutations, which is also located on the X chromosome. But now the CDKL5 deficiency disorder has its clinical identity. Now it is not covered under the Rett syndrome variants, because the major difference between these two diseases is, in case of the CDKL5 deficiency disorder, the kids have drug resistant seizures really early in the age, for example, even before 3 months of the age, whereas in case of Rett syndrome, the seizures arise after 3 years of their age. Moreover, the Rett syndrome has a normal phase of development. Children acquire normal crawling and speaking, standing skills. Whereas in case of CDKL5, the patients have no period of normal development. In case of Rett syndrome, they have the normal development phase, but which is followed by a rapid regression phase. They already start to lose their abilities that they have previously acquired. Whereas in case of CDKL5, as I told, there is no period of normal development, so they don't really acquire the basic skills that child acquire in their childhood. Now, the CDKL5 has its own clinical identity, and it is known as CDKL5 deficiency disorder, which is again a rare disease which is X-linked, as the gene is located on the X chromosome. And this disease is more rarer as it affects one person in around 40,000 to 60,000 live births. Unlike ASD, it influences females more than males, and the major symptoms of this disease are intellectual disability. There are very early onset drug resistant seizures. The patients have autistic like traits, as you can see this girl, which has this stereotypic hand movement. They have hypotonia. There are sensory, auditory, and visual impairments. Moreover, the patients have sleep disturbances. So, talking about the CDKL5 gene, it is a serine threonine kinase, and the kinase site is located on the N-terminus domain of the protein. There are various different mutations which occurs in the patients, but majority of these mutations end up with non-functional or unstable CDKL5 protein, which leads to the disrupted normal brain development. And the CDKL5 isoforms are differentiated by alternative splicing, and we know that the CDKL5-1 isoform is highly expressed in the brain of mouse as well as human beings. The CDKL in the brain, CDKL5 is abundantly expressed in the hippocampal and cortical regions, followed by a decreased expression in the thalamus and striatum. It starts to express immediately after birth at around postnatal day 2, and it peaks at around P14 in the mouse brain. The CDKL5 is expressed in neurons, where it plays differential role in cellular compartments in the nucleus. It phosphorylates various epigenetic factors such as DNMT1, MACP2, and so acts as a modulator of epigenetic factors. On the other hand, in the cytoplasm, it phosphorylates various microtubule-associated proteins such as EB2, MAP1S, and various other factors. So it acts as a regulator of dendritic and neuronal morphogenesis. Moreover, at the postsynaptic compartments, it phosphorylates NGL1, and so it modify various scaffolding proteins, and so acts as a regulator of synaptogenesis. So we know today various functions of CDKL5 in neurons, but still there is no cure for this disease. And to look for a cure, it is very important to understand more what this protein do in the physiological condition. And to understand this, various mouse models has been developed. We are also working with the transgenic mouse model, which is created by the deletion of exon 4 in the CDKL5 protein. And this model has been developed in 2014, and until now, in a decade, a very deep behavioral characterization of this model has been performed. This model has a high phase validity, and it mimics sufficient symptoms of the human pathology, such as the mouse show hind limb clasping, they have sensory and visual defects, there are defects in the learning and memory. The mouse also show autistic behavior. The only thing that we don't see in mouse is there are no spontaneous seizures as we see them in the patients. So using this mouse model, today I want to talk about metabotropic glutamate receptors. So because the mGluR receptors are highly involved in the pathways which are altered in CDKL5, such as there are defects in synaptogenesis and dendritic morphogenesis. So talking about the mGluR5, it regulates intracellular signaling pathways which control various physiological tasks, and it activates the translation of various proteins such as C-FOS and ARC, at least through two major signaling pathways, that is PI3K-AKT pathway and ARC pathway. And so it is involved in the expression of these proteins. On the other hand, the activation of specifically mGluR5 receptor, it modulates the functioning of NMDA receptor, and moreover, the mGluR5 is known to be essential to interact with OMER. So the OMER, if it interacts with OMER, it holds the receptor on the postsynaptic sites, and it is also important to exert the various functions of postsynaptic density proteins. So as we know that these are the major functions of this, so next we want to understand if the loss of CKL5 can interrupt the expression of these proteins. So we first looked at the CFOS, which is an immediate early gene. We performed this experiment in the primary somatosensory cortex, and as you can see from these images, there is a strong reduction in the expression of CFOS in the CDKL5 knockout males as compared to the wild type animals. Then next we looked also for other proteins that are known to be interactive with the mGluR5, that is postsynaptic density protein 95. And as you can see from these images, from this immunofluorescence images, there is a strong decrease in the expression of PSD95 as well as in the expression of OMER in the layer 2, 3 and layer 5 of the cerebral cortex. Next, we want to understand if there are any defects in the interaction of OMER1-BC with mGluR5. So we performed the co-immunoprecipitation and of OMER1-BC, and we performed the Western blot to label mGluR5, and as you can see in the wild type there is a strong interaction between OMER1-BC and mGluR5, whereas which is significantly decreased in the knockout animals. Then we next want to understand if there are what about the expression of mGluR5 itself in these transgenic animals. So we perform the immunofluorescence with mGluR5 antibody, and as you can see from these images, there is a strong decrease in the expression of mGluR5 in layer 2, 3 as well as in the layer 5 of the cortex. So all these results suggest that there are some abnormalities in the mGluR5 expression in these in these transgenic animals, as well as the functions that are modulated by mGluR5. Moreover, in our lab, we have the postmortem brain of 2 CDD patients, which are the only brains available worldwide for the moment. And we performed in the barrel cortex, in the B17, that is visual cortex. In the visual cortex of these patients, we performed the similar experiment. We looked for the expression of OMER1-BC, PSD95. Moreover, we also looked for the vGlut1, and we found that there is a strong decrease in the expression of these proteins in the patients as compared to the controls. We also have the selective controls for these relative controls for these patients, but we cannot perform any statistical analysis because the number of patients that we have is only 2. But as you can see from here, there is a strong tendency of decrease in these proteins in the patients compared with the neurotypical controls. Then we also performed the Western blot in the visual cortical lysates in the patients as well as controls. And as you can see, even from the Western blot, we found a decrease in the protein expression of PSD95 as well as mGluR5 and OMER1-BC, as you can see also from these bar graphs. So these results suggest that there is aberrant expression. The expression of these proteins is disrupted in the visual cortex of the CDD patients. So based on all these results, next we want to look for a therapy. We was looking for a druggable target, and as we know that mGluR5 can be used is a druggable target, and we can modulate the signaling of mGluR5 by using different drugs. So if we use any orthosteric agonist or a neutral agonist or a neutral or normal agonist, there are chances of the receptor sensitization, which leads to decreased therapeutic efficacy. So we looked, but it is possible to modulate the mGluR5s at the allosteric sites. And various studies have used positive or negative allustrators for allosteric modulators of mGluR5. In this study, we was interested to use a positive allosteric modulator. And so looking for the positive allosteric modulators for mGluR5, we end up with CDPPB, which is a drug that is known to be useful and that is known to rescue various behavioral and other glutamatergic defects in other diseases such as in SHRNK3 model and also in Huntington's disease. So we used CDPPB and we injected our mice with CDPPB, and 2 hours later we performed the behavioral and other visual and sensory motor analysis. So first we investigated the cortical visual responses in these mice before and after the injection by using we performed this analysis by using transcranial intrinsic optical imaging. And as you can see from these images, here in the CKL5 wild type animals, there are no alterations before and after the CTPPB treatment, whereas as you can see here, there is a rescue in the cortical visual response. There is an increase in the cortical visual response after the injection. Then we also performed other behavioral experiments. For example, we performed adhesive tape removal test in which a tape is applied on a normal tape is applied on the paw of the animal, and we observe the time when it starts to lick and it understands that there is tape on its hand, which is a test to understand the sensory ability of the animal. And as you can see here, in the CKL5 knockout animals, there is a strong increase in the time of the animal to contact the tape by licking or by observing it. Whereas when we treated these mice with CDPPB, there is a rescue of this phenotype. We also performed the Y-maze test, and we found that there are strong alterations in this behavior in the knockout animals, which was significantly rescued by the CTPPB treatment. We then also next we want to look for the other synaptic and postsynaptic density proteins, and as well as for the directly for the mGluR5 receptor expression, and we perform the immunofluorescence for this. And as you can see from these images, this decrease in the OMR1BC expression was significantly rescued in the layer 2, 3 and layer 5 of somatosensory as well as visual cortex in these mutants, as compared to the animals that was treated only with the vehicle of the wild types. Moreover, there is a rescue in the expression of mGluR5 as well, as you can see from these graphs. And we also look for the expression of ARC, which is an early immediate gene, as I explained before. Also for this protein, there is a rescue by the CDPPB treatment in the mutants as compared with the vehicle-treated mutants. Then next we want to we used another mGluR5 PAM, which is called Ro68, and this drug is more potent than CDPPB, and it has a low toxicity because the CDPPB is dissolved in DMSO, whereas this drug is dissolved in just 0.3% of twin80, which is really a less toxic vehicle. So we try to use also this compound, and with this compound as well, we perform the similar type of dose, and we performed the intraperitoneal injection, and we analyzed different parameters after 2 hours of the treatment. First, we analyzed the CFOS, and as we saw before, there was a decrease in the CFOS expression, which was significantly rescued by the expression by the CDPPB treatment in these mutants. And this rescue we can observe in almost all cortical layers of the somatosensory cortex. Then we also look for the expression of OMR1BC again with this new drug, and this drug also seems to rescue even with very low dose in the for this expression of OMR1BC in the mutants. So to sum up my talk, I would like to say that what we understood from this study is that mGluR5 synaptic localization is decreased along with the reduced mGluR5 mediated signaling. Moreover, the loss of CKL5 tampers the binding strength of the OMR1BC mGluR5 complex. So probably what we understood is the mGluR5 do not bind as prominent as it can bind with the with OMR1BC presence to the postsynaptic sites. Moreover, this study also tells us that mGluR5 is a putative therapeutic target for preclinical pharmacological treatments for the CKL5 deficiency disorder. And at the end, I want to thank all my colleagues, my professor Maurizio Giustetto, my seniors Riccardo and Antonia, and all my colleagues who helped me during this work. And thank you all for your attention.
Thanks a lot, Dr. Sunaina, for a wonderful talk and wonderful lecture. You have started your lecture with autism spectrum disorders, then you have explained about its variants, then you have explained about Rett syndrome variants, then CDKL5 deficiency disorder, CDKLT gene and its roles. Also, you have explained about CDD mouse models and mGluR5 as a potent target against uh, these particular diseases. And you have also told the drug about CDPPV, which is a potent therapeutic candidate in this disease. So I just want to know that, Sunaina, is CDPPB and the other drugs, these are developed by your lab or you have tested them from another something?
Yes, so the CDPPB is a formulated drug, and you can find it in the chemist stores or in some other. I mean, it's a non-drug.
It
It
is
is also
FDA
used for
approved
other
already? It
things.
is available in market?
Yes, not for this disease, but for some other disorders. It is a
Okay.
formulation that you can easily find from some companies and other things. Whereas the other drug that I was talking about, Ro68, it is a compound that is developed by a lab in the lab of Professor Claudio Supuran at the University of Florence. We have not created it. We have not formulated it by ourselves, but we have obtained it from a collaborator that is from the University of Florence. They have formulated this drug by themselves, and you cannot find it until now somewhere else.
So, being a medicinal chemist, how can I explore this target against other diseases? Yeah, is this target involved in other diseases rather than this?
Actually, these days, mGluR5, even not only mGluR5, but also other subunits of metabotropic glutamate receptors are really involved in various other diseases, such as they are also known to have function in demyelinating diseases, also in various neurodegenerative diseases too, not only neurodevelopmental disorders. Already in various neurodevelopmental disorders, not only in autism or CDKL5 as we tested, but in other neurodevelopmental disorders, they have been studied, and they have known to show positive effects. So if in a disease there are defects or abnormalities in the expression of mGluR5, or if there are the down targets of mGluR5 that are altered, it could be a very good idea to use it as a druggable target. Yeah, because
Okay.
this is something that you can target, because you cannot always target all the receptors or proteins that are present in our brain, but mGluR5 is a druggable target, and if the things that go wrong in the downstream of this receptor, it could be very useful to target mGluR5s.
And what's your opinion about clinical success of these studies?
So we have tried this drug in two of the post-mortem brains. As I told you, we have two post-mortem
Okay.
brains from CDD patients. So we look
Okay.
forward for this, but still there is a lot of work which is need to perform in mouse models, or even in the post-mortem brains in the CDD patients' pathology. Then maybe they can go to the clinical trials. But already, not for this disease, but for various other disease, mGluR5s or even metabotropic, other subunits of metabotropic glutamate receptors have been gone to the clinical trials, and they are known to be successful as well for some other diseases. Some of them are in phase 3 trials already, so could be very, we are looking forward to have some positive results for this.
Okay. Thank you, Dr. Sunaina. You have definitely provided us a new target. So, as being a
Yeah.
medicinal chemist, we can also go for in silico studies and design some compound against them. So, thank you so
Yes,
much for your
yes.
wonderful lecture. Now, over to
Thank you
our
so much for
respected
this
director, sir.
opportunity. Yeah.
You are welcome. Over to director, sir, for
So,
saying some words.
many excellent, Dr. Rohitji. So, Sunaina deliver a very excellent talk, very precise, very specific as we are expecting. So, I hope that everybody will benefited with the talk given by the Sunaina, because I know that when the people is moving from the India to the abroad, they have to the lot of change there in the drug delivery system, as well as the way of presentation. They are very precisely working over there, and they are specifically work on the particular target. And I hope that this types of the talk it will be the useful to the youngster, because sometimes we are the diverted the so many field, so we need to the work on a specific field as well as the target. So thank you very much, Dr. Sunena. And as and when you visit the India, or you feel that okay, we can some do the work with the some collaboration, because ISF College of Pharmacy now growing in the very fast track in the research, and we are also involved some of the business incubation center as well as some of the testing. Now we are the moving toward the startup and entrepreneurship, because after all, if we want to the establish our self as a researcher or research institute, then we need to some of the entrepreneurship and innovation is the compulsory. So I hope that your talk, your interaction, your involvement, it will be the beneficial to all the person, those who are the connect through the direct Google Meet as well as with the YouTube channel. Now my another request, if you have want to discuss, or you want to give some name of your team member, those who want to interact or involve with this ISF dialogue series, you can also provide, so that Dr. Rohit ji connect these peoples, because it is not essential that we can connect only some of the person. So some of the Italians and some person they want to deliver their own work, so they are welcome is also. So thank you very much, and
Yes.
God bless
Sure.
you. You can grow very fast, and this is the proud moment to also that one of my alumni in front of me and delivering one of the scientific talk. So you can say some number of time I mentioned that कि hemoglobin बढ़ जाता है. So this is the moment to me. Thank you very much. God bless you.
Thank you so much, sir. Thank you so much. Thanks a lot for
Thank
the
you,
kind
director, sir.
words.
Thank
So, Sunena, our a few pharmacologists they want to interact with you, so I will share your email ID with them. So, if
Yes,
they
sure,
need
sir.
any
Yeah.
kind of opinion from you, just help out them.
Sure,
So,
sure. That will be my pleasure.
warm wishes for your postdoc, and thanks a lot again.
Thank you so much. Thanks a lot. That's so nice to see you after so long, and that's so it's so good to interact with our professors and our teachers from who we teach, who from whom we learned a lot. And today I'm here, so I'm so thankful of you, and of course to the SF College. Thank you so much.
Thank you, Sunena.
Thanks
Okay,
a
thank
lot.
you. Bye.
Bye. Thank you. Have a
Bye
nice
bye.
day. Bye.